Welcome to Reviewing with Mrs. Wages. Today I'm reviewing cell transport. Objective number 1 asks you to describe the structure and function of the cell membrane. The bulk of the cell membrane is made of phospholipids. These molecules have a phosphate head that's polar, making it attracted to water. Each phospholipid also has two lipid legs, which are nonpolar, meaning they repel water. The phospholipids arrange themselves with the lipid legs pointing together, forming a bilayer. The nonpolar lipid legs form a barrier region, allowing the cell membrane to do its job, which is to control what goes in and out of the cell. There are also various types of proteins embedded in the phospholipid bilayer. One type is a channel protein, which is important for moving certain materials across the cell membrane. If a molecule is small enough and nonpolar, it can cross the fluid cell membrane by wiggling between the phospholipids. But if a molecule is too large or polar, it cannot. Remember, the lipid legs form the barrier region of the membrane. If a polar molecule tried to cross, the nonpolar lipid legs would reject it. This means that the polar molecule and the large molecules need a different way to cross the membrane. They need to use protein channels. Protein channels, or channel proteins, are like secret passageways for the large or polar molecules to cross the membrane without being rejected by the lipid barrier region of the bilayer. Now let's talk about types of transport processes. Some processes require energy, others do not. The focus of objective number 2 is passive transport. You learned about 3 types of passive transport. In each of these, molecules are moving down the concentration gradient. In other words, the molecules are moving on their own from areas of high concentration to low concentration. Because of this, the cell is using no energy to move the molecules. In diffusion, the solute molecules passively move from high to low concentration to try to reach equilibrium. This can happen with or without a cell membrane. Atoms of a liquid are continually moving and colliding into one another, and because of this, they tend to spread out evenly if possible. This would be equilibrium. The same thing happens in facilitated diffusion, except for the cell helps by providing a protein channel, but it still does not use any energy to move the molecule. The molecules just collide their way through the channel. This picture shows facilitated diffusion because the solute particles are moving through a protein channel from an area of high concentration to an area of low concentration. Again, the cell is doing no work. In osmosis, the same thing happens, but with water. And remember, water is a solvent. During osmosis, water will move from areas of high concentration of water to areas of lower concentration of water. This is the same thing as saying that water will move via osmosis to the hypertonic side of the membrane. When trying to predict which way a molecule will move, it's critical to consider the concentration rather than the number of molecules in an area. In this example, let's predict which way the solute will diffuse. Inside of the cell, we have 3 grams of solute. We also have 3 liters of solvent. Outside of the cell, we have 3 grams of solute as well, but we only have 2 liters of solvent. If I reduce the ratio, you can see that I have on the inside the concentration of 1 gram per liter. On the outside, I have 1.5 grams per liter. Clearly, 1.5 is greater than 1, therefore outside is considered to be hypertonic, which means more solute, and inside is considered to be hypotonic. Since this is diffusion of the solute, we can then predict that the solute will move from high to low concentration, meaning it will move from outside to inside of the cell. In this example, let's predict which way osmosis will move water. Here you can see that inside the cell, we have 4 grams of solute and 3 liters of solvent. If I reduce that ratio, our inside concentration is equal to 1.3 grams per liter. Outside, I have 6 grams of solute and only 3 liters of solvent, making my outside concentration 2 grams per liter. Water is going to move towards the hypertonic side because if it's hypertonic, it means it has a lower concentration of water. Therefore, water, which is the solvent, will move from the inside to the outside of the cell. In objective number 3, you are to describe various types of active transport. The 3 types of active transport that you learned about are the pumps, endocytosis, and exocytosis. In this example of active transport, you are seeing solute being pumped using energy in the form of ATP to an area of higher concentration, which is against the concentration gradient. We're going from hypo to hypertonic situations using energy. In endocytosis, the cell uses energy to reach out and engulf material to bring it inward. Engulfing solid food, phagocytosis, and engulfing liquids, penocytosis, are both examples of endocytosis. During exocytosis, vesicles carrying material merge with the cell membrane and the material is released. An example would be when a cell releases proteins from the vesicles produced by the Golgi bodies. And because it's active transport, exocytosis requires cell energy. Now let's flash back to monomers and polymers. Monomers are small building block type molecules that combine to form macromolecules called polymers. Polymers, then, are those macromolecules composed of many monomers of the same type. Proteins are polymers. Some of them, like enzymes, control the rate of chemical reactions in cells. The amino acids are the monomers of any protein. Nucleic acids are polymers. Nucleic acids are heredity molecules found in cells. They store and transmit genetic information. Examples of nucleic acids are DNA and RNA, which means both DNA and RNA store and transmit genetic information. The monomers of any nucleic acid would be a nucleotide. Polysaccharides are polymers and are the main source of energy for most living things. Monosaccharides are the monomers of polysaccharides. That's all for now. I hope this helps, and as always, happy studying!